Thank you for the introduction and good morning everyone. So let's talk about hemp. So hemp is considered as a sustainable crop because of its multi-purpose use and it has high potential for carbon sequestration. So hemp is mostly grown for grain, and also it's grown for fiber, and it's mostly popular for CBD or pharmaceutical properties. So there is lots of byproducts in hemp industry. If we consider the CBD hemp or plural hemp, there is some byproducts or spent biomass. Those spent biomass have been evaluated for potential animal feed, but we also can consider hemp as a forage, but there is a limited study which conducted hemp as a forage. So there is also the lots of interest on hemp to consider as a forest crop. So that's why we conducted that study. So in our study, we try to address a few of the questions. So one is if we consider hemp as a forest crop, so then we need to evaluate what is the nutritional value and what kind of cultivar we can utilize for forest crop. So that's why we are trying to address one question, that is nutritional difference between hemp cultivars, and also the hemp is kind of photoperiod sensitive crop, and it's if we consider this hemp as forest crop, and harvesting time may be an important issue. That's why we wanted to address that issue, which is nutritional difference between harvesting time. So to address those questions, we conducted that study at Virginia location, the Virginia Blacksburg location. So for that study, we evaluated two cultivar. One is called Grandi, which is known for grain hemp, and another one is Joey, which is known for dual purpose. That means we can cultivate that Joey cultivar for grain and fiber both way. So there is some architectural difference between those cultivar. So to conduct the study, we planted those two cultivar with conventional tillage and with 112 kilogram nitrogen per hectare, and seeding rate was 34 kilogram per hectare, and we seeded on May 13, 21. We conducted that study in 2022 as well, but here I am presenting only data from 2021. So we harvested those samples at 3 weeks after interval. We used a quadrate to harvest the plant material, and we harvested the whole plant at 5-inch height. And after that, we processed those sample, and then we analyzed using NIRs, using mixed hay calibration, and then we analyze using mixed effect with repeated measures using SAS Studio. So now I'm going to the results. Here you can see this data is for the plant height. So we we observed there was interaction between harvest time and cultivar, and that's why here this data is analyzed separately by cultivar, and this Grandi and Joey we analyzed separately. So if we consider the cultivar Grandi, we didn't see any difference after June 14. That means after that they didn't grow vertically. But Joey, that is another cultivar which we seed after June 28, we didn't observe any increase of plant height. So that means these two cultivar is morphologically different, and their architecture is different. However, in the next slide, we can see the biomass yield. We didn't observe any interaction between harvest time and cultivar. That's why these data are average across two cultivar. So from this figure, we can see the biomass yield increased steadily, though there was difference between height, but biomass yield was same. On July 19, we see, sorry, maybe I can use this one. So the July 19, at the end of the harvest, we observe highest yield, that is 3.1 megagram per hectare. So if we consider this hemp as a forest crop, maybe this yield is very low. But if we consider the time frame, as we planted May 13 and harvested until July 19, that means around 2 months after establishment. So in that way, if we consider within 2 months, we can get around 3 megagram per hectare yield. That is maybe potential, but if we consider overall biomass yield, it's kind of low biomass. So in this figure, we can see the nutritive value. So evaluated crude protein content, ADF, NDF, and lignin. So if we consider the crude protein content, which represent the green color, so at the first harvest, we can see on June 7, crude protein content was 31%, and low crude protein content was observed on June 28, which was 19%, which is also still high. So here we can see the crude protein content in hemp is really high. So as I've said earlier, we applied 112 kilogram nitrogen. Though we didn't analyze nitrate content in the plant tissue sample, these are crude protein content. Maybe there is some effect from nitrate, but we analyzed in wet chemistry as well. We also found around in whole plant around 25 to 26% crude protein content. At the early stage, crude protein content is high because the leaf content is high. And if we consider the ADF and NDF, we see at the early stage their ADF and NDF content was low, around 22%, and it's increased 35 to 45% at the later stage, which is expected as plant matured. And on the bottom one, like in black color, represent the lignin content. We see at the early stage lignin content was low, which is around 4%, and at the later stage it's increased to the until July 19, we observed 9% lignin content, which is expected as plant matured, lignin content become high. So this figure, we can see here is the total digestible nutrient, and from the left side figure, we can see the TDN content was variable at different harvesting time, and we didn't observe any the interaction between harvest time and cultivar. That's why these data are average across two cultivar. So generally, if it is 50% TDN content is considered adequate for non-lactating cows. So here we can see at all harvest time, its content was more than 50%, but yeah, more than 60%. So which means digestive, in the standpoint of digestibility, its digestibility is high. But on the right side, you can see the TDN yield, as total biomass yield was low. So here, that's why here TDN yield also low. And highest TDN yield was observed around 1.9 megagram per hectare at July 19th. So these are some data. So based on this preliminary data, we can say nutritionally hemp has promise as a forest crop. But if we consider the biomass yield and total production, which maybe we need to evaluate further with the different cultivars. And another thing, we didn't analyze or didn't conduct any study like that, animal intake study. So in future, if we want to consider hemp as a forest, we need to conduct some studies like which can address animal intake and performance. and also we need to address establishment and also production management with different cultivars at different stages. So that's it. Thank you all, and if you have any question, let me know.
Thank you, Charles. We have plenty of time for questions. Any question?
Yes, sure.
If you're drug seeding this, and how would you, thinking of animal intake, how would you anticipate animals intaking this? You releasing it on or harvesting it and baling it? Like, if you're looking for research on animal intake, how would you suggest that take place?
So based on my like observation, if you harvest early stage, you can bale it, and but we didn't conduct that study. And also maybe it's like my personal observation. Maybe if you want to graze with the other grass, you can mix with that at early stage. But at the later stage, plant becomes so thick or more fiber. So maybe palatability will reduce maybe.
One more question?
Hi, thank you for your presentation.
Yeah, thank you.
It's interesting to know that there's new options to feed animals. I wonder if you can give us some indication about future research on animals' performance.
Yep.
If there's any clue that you can
We didn't conduct any of the animal intake, but there is some study. They conducted study on spent biomass or like byproduct from the CBD hemp. Like from the CBD hemp, after extracting the oil or CBD oil, then there is some biomass. There is some study they conducted that one, and but only for the forage, hemp as a forage, there is no study. But I think we need to evaluate that.
Are there any animal welfare concerns or regulatory?
Yeah, right now the hemp is not like legalized or authorized to feed in US. So that's why we need to conduct more research study to see if there is any like a negative or positive effect if we feed a hemp or hemp byproduct.
More questions? What other species can you grow in the same area? And what is the yield potential of the other species?
Other species, other cultivar?
No, other species, not
Oh,
cultivars.
yeah, other species, there is like, if you consider warm season, there is Sudan grass, their potentiality is so high, like biomass yield is so high, and the alfalfa is a widely known forest crop. If we consider the nutritive value, it's look like similar with alfalfa, but alfalfa is kind of perennial crop, but here this hemp is annual crop, so we need to plant again. So there is one issue.
Okay. One more question?
Yeah, sure.
About your varieties that you chose, were those only two available to you for industrial production?
No, there is some other cultivar as well, but these two are widely known for grain and dual purpose. So but there is other cultivar, so we can evaluate those as well.
Okay. We are out
Okay.
of time. Thank you very much.
Thank you so much.
Okay, our next presentation is titled Plantain, Plantago lanceolata. Growth is limited under water logging. The presenter is Sam Wilson. Yes.
All good. Good morning, everyone. I'm Sam. I'm a PhD student at Massey University in New Zealand. My PhD thesis title is investigating the impact of abiotic stresses on the ability of plantain to reduce nitrogen losses from grazed pastoral systems. The experiment that I'm presenting to you today is part of my first research objective, which is to investigate the impact of waterlogging on plantain, and that was done in the glasshouse. So for those who aren't familiar, this is what New Zealand looks like. And the photo in the background was taken on my parents' farm near my hometown of Hunterville, which is under the red arrow there on the map. The farm is mostly sheep with some cattle, and it's the main farm type in the area. 45 minutes southeast, 45 minutes drive, is the Manawatu region, which has a high concentration of dairy farms, and it's also home to Palmerston North, Massey University, and my PhD research. The New Zealand economy is built on the back of pasture production, which pretty much occurs in all of these lighter green areas around the map here, and that's reflected in New Zealand's annual export earnings, with last year dairy products accounting for 25% of exports and meat products accounting for 8% of exports. These sectors rely on high quality, low cost pasture based inputs to remain profitable, and there are several challenges associated with that year round pasture production. Now some other more recognisable products of New Zealand. Here we have the All Blacks, the national rugby team, arguably the most successful sports team in the world. Then we have Stephen Adams, our most famous basketballer you might recognise, and Taika Waititi, our most famous member of Hollywood at the moment. So one problem with year-round pasture grazing on high-quality ryegrass white clover pasture is the pollution of freshwater resources with nitrogen. When cows eat the pasture, the nitrogen content of the feed far exceeds the nitrogen requirement of the grazing animal, and so most of the nitrogen is returned to the pasture via the urine. In these urine patches, there is so much nitrogen that plants are not able to take it up before it's lost to deeper portions of the soil profile and into fresh water. The result of excess nitrogen, along with excess phosphorus from sediment, is eutrophication and algal blooms. These essentially work to exhaust the oxygen supply of the waterway, leading to the death of fish and invertebrate species and a loss of biodiversity in the waterway. Additionally, freshwater sources can become too high in nitrates for safe human consumption, which poses a risk to the water security of local populations. So one simple and low-cost solution to this problem is plantain. Now, I'm not talking about bananas. Plantago lanceolata, also known as narrow-leaved plantain or ribwort plantain, is a deep-rooted perennial herb or forb species capable of higher growth rates during summer months, higher growth rates than other typical temperate pasture species, but it does have low cool season activity and can be sensitive to close grazing and treading damage from livestock. One of the special features of plantain is that it contains secondary metabolites that, when eaten by cows, can reduce the concentration of nitrogen in their urine and increase urine volume. The result is a change in the way that nitrogen is spread over the pasture. On the left, we have a traditional ryegrass white clover pasture high in nitrogen. You can see there's few urine patches with a high concentration of nitrogen in each patch. And on the right, in the plantain pasture, there's more urine patches because the cows need to urinate more often, and there's less nitrogen in each patch. In the plantain pasture, plants can take up more of the nitrogen before it is lost to deeper portions of the profile and soil water, leading to a reduction in nitrogen losses from the system. The same compounds can also reduce nitrogen leaching potential further by suppressing nitrification in the soil, which reduces the amount of nitrate, which is the readily leachable form of nitrogen. For plantain to produce meaningful reductions in nitrogen losses from farms, it needs to make up at least 30% of pasture yield. There is evidence that plantain has poor persistence in New Zealand dairy pastures. This data from New Zealand dairy farms suggests that plantain contents greater than 30% are only attainable in the first 2 years following the drilling of the pasture, and cannot be maintained thereafter. Competition from other pasture species, notably perennial ryegrass, overgrazing, treading damage and compaction are all causes of this sharp decline. There is also evidence that plantain does not cope well in waterlogged soils, which occur on many New Zealand dairy farms every year. However, there's very little previous research that has considered the effect of waterlogging on plantain, so this study helps to try and fill that gap. The aims of the experiment were to investigate the impact of waterlogging on plantain growth and then compare the responses to waterlogging of plantain and perennial ryegrass. Water treatments were defined by soil volumetric water content as follows. Control pots were kept at an optimum soil water content just below field capacity. Wet pots were kept above field capacity but not at saturation, and waterlogged pots were kept saturated by placing them into larger tubs so that the water table could be maintained at 5 centimetres below the soil surface. The water treatments were imposed for 39 days total before 27 days of recovery. There were leaf harvests at 22 days, so a mid-stress harvest at the end of the stress and then following recovery, and roots were harvested also at the end of the stress period and at the end of the recovery period. So a couple of graphs here that show leaf dry mass production by the two species. So on the top we have leaf dry mass production under the stress period. So that includes both of the days 22 and 39 harvests. And then we have leaf dry mass produced during the recovery period. The plantain is in the yellow, perennial ryegrass in the blue, and the order of treatments is always control, wet, and waterlogged. So waterlogging reduced the leaf growth of both species during the stress and recovery periods. Plantain leaf growth was reduced by 37% under waterlogging during the stress period and 38% under waterlogging during the recovery period. That indicates that plantain leaf growth is sensitive to waterlogging stress and that previously waterlogged plants may take an extended period to recover. However, no plantain plants died under the waterlogging, which suggests that it does possess some waterlogging tolerance strategies. In contrast, perennial ryegrass leaf growth was only reduced by 18% by the waterlogging during the stress period, and it was able to match control plants during the recovery period once the waterlogging stress had been removed. That suggests that perennial ryegrass has a good degree of waterlogging tolerance, and that agrees with previous studies. So when we compare the two species, perennial ryegrass produced 28% more leaf dry mass under the stress than plantain, and 45% more leaf dry mass than plantain during the recovery period in the waterlog treatment. That suggests that perennial ryegrass has an enhanced capacity for coping with and recovering from waterlogging in comparison with plantain. So this is the root dry mass data. Again, we have plantain in the yellow, ryegrass in the blue. On the top is root dry mass harvested at the end of the stress period, and then root dry mass harvested at the end of the recovery period. Perennial ryegrass root dry mass was 33% higher under waterlogging than the control, which agrees with previous studies. In contrast, increased root growth didn't appear to be a waterlogging tolerance strategy of plantain, but root dry mass was maintained in plantain under waterlogging, and that suggests that either root growth continued as normal with the aid of an extra oxygen supply from the surface, such as something like erythema, which is air transport cells, or plantain plants developed adventitious roots closer to the soil surface to compensate for a loss of roots deeper in the soil in the flooded zone. And you can kind of see some of that latter effect happening in this picture. It's not that clear, but so both of these have the same mass, but you can kind of see on the right there's a increased number of thickened roots nearer to the base of the plant in the waterlog treatment. So to conclude, plantain growth was sensitive to waterlogging in this experiment. However, the survival of plantain under waterlogging suggests that it does possess some waterlogging tolerance strategies. In contrast, perennial ryegrass proved to have enhanced capacity for coping with waterlogging. The findings suggest that plantain growth may be limited in waterlogged ryegrass-based pastures, and the enhanced ability of perennial ryegrass for coping with and recovering from waterlogging could provide it with a competitive advantage over plantain under such conditions should they coexist in a mixed sward. So thank you for listening, and I just need to make mention of the TR Ellett Agriculture Research Trust for project funding and the School of Agriculture and Environment for getting me to the conference. Thank you.
We have time for 2 questions.
So, question of the targets for plantain incorporation in the system, and what's effective in terms of changing the nitrogen in the system? I mean, how much plantain do you need in a pasture?
Yeah,
Let's say a benefit relative to the environmental impact.
the early evidence, the figure that we work on is 30% of the pasture dry mass, which is based on a couple of studies from New Zealand. So roughly 30% of the pasture dry mass, which means that you would be also feeding 30% plantain dry as a percentage of the diet to the cows to get the reduction in urine nitrogen concentration and the increase in urine volume as well. So 30%.
So the second question then would be strategies, and you've got this decline in plantain populations over time. What are your strategies for maintaining that, or
Yeah.
is it just a reseeding issue, or how do you do it?
At the moment, the only strategy is to kind of oversow it, direct drill it every 2 or 3 years once you see it drop below that 30%. Usually farmers will make that assessment based on a visual assessment to make sure it's at 30%. So it's mainly at the moment reseeding, but my kind of thinking is let's try and find alternatives to try and keep the plantain content higher, because reseeding all the time can get reasonably expensive for farmers, I think.
Sorry, we don't have time for more questions. Thank you, Sam.
Thank you.
Well, I welcome Luis Nostroza. He will present the paper Root and Above Ground Traits Expressed by Land Races and Interspecific Hybrid of Alfalfa with Putative Drought Tolerance in Mediterranean Environments.
Morning, everyone. Thank you so much for the introduction. Chile is one of the 5 regions across the world that has a Mediterranean climate. A Mediterranean climate has the characteristic that with a cold and raining winter, with more than the 80% of the rainfall is concentrated during the winter period, and also has a hot and dry summer. This is providing almost 5 months or more than 5 months of negative water balance with a lasting drought period. So across the world, Mediterranean environment are one of the most threatened by the climate change condition. So any decrease in rainfalls is affecting the entire farming system and their profitability. In Chile, as you can see in this plot, the red dots are the drought period in the last century. So historically, in Chile, we observe 2 or 4 years of drought every decade. So in the last decade, the entire, every year of the decade has been in a drought condition. So this situation or this condition has been called the mega drought period in Chile, and this condition is affecting all the farming system developed in dryland areas. So as a strategy, we are proposing alfalfa as a genetic resource for adapting to livestock system to climate change. Alfalfa has several benefits. Among them, as you know, alfalfa has a great agronomic value with a high forest production, high forest quality, and also has several ecological services, like nitrogen fixation and so on. Additionally, it's called tolerance for our environments, but the most important trait is the drought tolerant, because as you know, alfalfa has a deep root system and also have other physiological traits that are conferring alfalfa a greater adaptation in Mediterranean environment. So through the alfalfa crop wild-related program supported by the Crop Trust, we introduced to Chile 69 alfalfa accession, and we phenotypically characterize this alfalfa diversity panel in Mediterranean environment of Chile and also in Australia. In this table, you can see the original country of every accession, and this accession belongs to 4 Medicago sativa subspecies. Among them, we introduced 2 hybrids of Medicago alborea, 2 Medicago caerulea, 50 Medicago sativa, and 14 Medicago varia. This is a picture of the Mediterranean environment as we are working. In the spring period, as you can see, everything is green. As soon as we get summer, the situation is totally brown, except the forest area, the vineyards, and today, the alfalfa area. As I've mentioned before, we characterize phenotypically this 69 accession in Chile and Australia, and we select some drought-tolerant accession. And the drought-tolerant was considered based on their agronomical performance in this condition, with 5 or 6 months of drought period. And now we are interested in understand or illustrate what have this drought-tolerant accession that are able to survive more than 6 growing seasons in this kind of environment and also produce biomass. So the objective of this study was to evaluate the below and above ground phenotypic expression of drought-tolerant alfalfa accessions in order to identify morphophysiological mechanisms conferring to alfalfa greater agronomical performance in Mediterranean environment. We work with 8 alfalfa population, 4 landraces originated from Chile, Spain, and Peru, 2 Australian cultivars, 2 Alborea hybrid, and we established this accession in PVC tube of 1 meter depth, containing a substrate of sand, vermiculite, soil, and perlite. And plants were grown are two water regimes, with water deficits and well water condition. We organized the experiment in a RCV design with 5 tubes per replicate. So we were working with around 240 tubes in this experiment. We evaluate some physiological and morphological traits, phenotypic trait at 130 and 174 days after sowing. So as we expect, the drought condition, the water deficit, affects the entire phenotypic expression of this plant. The plant water potential and stomal conductance was reduced 3 times in water deficit condition related to the well water condition. Additionally, some leaf traits like a leaf area and a specific leaf area was reducing at 25%. So every modification at above ground level results in a reduction of the 40% in the biomass production, chute dry matter production. At population level, we observed that the Peruvian population, APG 44669, obtained the highest biomass production, and the Chilean accession, the lowest. But this biomass production was associated with, in the case of the lowest biomass production, the population obtained the highest stomal conductance, which was a little bit weird. And also, the population with the highest biomass production obtained the lowest stomal conductance. So, as I mentioned before, all these populations are drought tolerant, but they are expressing different phenotypic traits in order to fight or face with the drought condition. So we are so excited with the result because we have some difference at gas exchange levels that we are trying to still working on. At root level, root biomass production and partitioning across the tube's depth, we observed that some population, we observed a significant population by water environment interaction, and we observed 3 patterns of biomass production at root level and root dry matter partitioning in the depth. So the Chilean and Peruvian accession increased the biomass production when was growing at a water deficit condition. On the other hand, the Australian cultivars, they decreased their biomass production, the root biomass production in the water deficit condition. And the other accession, the Alborea hybrid, and the Spain accession didn't change their root dry matter production in the 2 water environment. So as a conclusion, we can mention that the alfalfa population with the putative drought tolerant in Mediterranean environment didn't exhibit a unique phenotypic strategy for facing severe water stress. And stomal conductance and leaf traits and root architecture and root biomass partitioning exhibit the most divergent phenotypic expression. Thank you so much.
We have time for 3 or 4 questions.
No question.
Why you were surprised that this semantical conductance was opposite in the production? I would say the conductance of somatic conductance is lower, but probably the plant is safe in water, and that might explain the high production.
Yeah. We have a lot of data about the stomal conductance in forest species, and most of the time we observe that the most of the time, shoot biomass is associated with higher highest stomach conductance. So we never expect these results. I know that there are some plants saving water, but this mechanism most of the time is associated with a lower biomass production.
One question about the methodology. Do you have one plant per tube?
Yeah.
So 5 plants per variety per
replicate.
one question.
The what?
Altitude elevation above sea level?
This was a greenhouse experiment, and we was working at 100 meters above sea level.
More questions? So how will you continue with this work?
Yeah, this work is belong to a large project of alfalfa breeding for Mediterranean environment. So this information give us some orientation about the phenotypic trait that we have to evaluate in the new variety that we are trying to release. Okay.
More questions? One more question?
I saw you collect a lot of trays, and I saw you working with the crop tracks. Are you feeding back the information that you measured to the database of the gene banks? So usually you can feed back with this information to characterize the accessions that you evaluate. So I wonder if you're doing that.
Yeah. So we are still working with the alfalfa curator from the Australian gene bank, and he's getting and collecting all this information. We are still working in a research project, so all this information will be available in the Green Global website. I don't know.
Okay, thank you very much, Luis. Well, we welcome David Mwangi, who will present the work evaluation of apomictic Magat Hirsus maximus accessions and ecotypes to improve feed availability in Western Kenya. Thank you very much.
Thank you. If we can get this working. Thank you, Chairman. I'll be very briefly talking about some work that we are doing with Megadaisus maximus accessions in Kenya. Those of us who might not have been to Kenya, in East Africa, we have a situation where most of our milk is produced by very small scale farmers, less than a hectare in size, but producing milk mainly based on fodder crops like Napier grass. But we have a few issues with that, and that's why we are starting to evaluate other materials that might help improve the fodder situation in some of these farms. Now, those smallholder systems are characterized by very low productivity of animals. So even where we have had increase in milk production, it has been more based on increase of numbers than really an increase in productivity. These animals that are capable of producing about, sorry about that, wow. Need to get this right. Try to get the pointer on this.
Okay.
Trying to get the pointer.
Forward?
Yep. The pointer.
It's here.
Okay. Okay. So very low productivity. These are mainly Frisian animals that should be able to give 4,500 liters lactation, but they're only doing about 1,400, mainly just because of feed issues. And as I said, Napier grass is one of the major feeds. But now we have a major problem with that. When you have vegetatively propagated fodder, then you have a very vulnerable population, and we have now 2 diseases in Kenya that are affecting Napier grass. One is a stunt disease caused by a mycoplasma spread by the leaf hoppers. And then we have Napier stunting disease that then reduces the biomass, this big biomass from these plants to negligible amounts. So therefore, we are looking for different materials that would replace Napier grass in those areas. So the approach we have taken 2 approaches. The first one is we are collecting and characterizing our own local materials and also breeding into that, but also repatriating some of the improved varieties of some of our grasses in the region back into the region and evaluating them to see how they would perform. And this is where our big connection with the Bracharia and the panicum systems that have been developed in Argentina and Brazil come back into our system. So now I'm working on Megathyssus maximus to look at whether it could actually replace this Napier grass that we have been growing before. Now, we collected about 36 accessions, 17 of these from the CIAT gene bank in Colombia, and another 16 collected locally in our area. And we tested them in Western Kenya in an area called Kakamega, near Lake Victoria, which is, that's the position of Kakamega. And the rainfall is about, this is about 2,000 millimeters of rainfall in a year. And we planted the material in plots in that area, and we had quite a number of materials in that. And anything that doesn't have a CIAT number on this means was collected locally, and we have several materials that are also commercially available in the area. So we looked at 2 major characteristics: the dry matter yield from the materials, but we also looked at the leaf stem ratio just to see a bit on the quality of the material that we had collected. Now, these 36 accessions were planted in single rows because we couldn't really replicate this because there were too many. But after that, we selected a number of them that then went into replicated trials. And some of the results that I'll be giving will be on the dry matter yields. So we had a range of yields, and I'll be showing a graph on that. And we had anything from about 12 tons per hectare of dry matter to about 6 tons of dry matter per hectare. This compares very well with what we get from elephant grass or Napier grass, which is about 14, 15 tons of dry matter. But we had several varieties or several accessions that had a bit higher dry matter than others. And these are listed here. MK6, this is a local collection, and these 2 are local collections, and the rest are CIAT materials. And then we have one taken from Brazil called Tanzania, and another one also taken from Brazil called Mombasa. And some of the locally collected materials are very similar dry matter yields compared with these materials that we have repatriated from Brazil. And what we are looking at right now is issues of persistence and disease and pests on these materials, because the moment you start repatriating materials, you want to see what the effect of disease would be. Now, we also looked at the leaf to stem ratio of these materials, and there was a very wide range of results from materials that had very, very high stem components, and therefore very few leaves, and therefore the quality very poor, to materials that had quite a bit of leaves, and therefore expected to have better nutritional quality for our animals. And now, based on this, we had a number of conclusions and a number of areas where we are going into. We had, we now think we could actually use Megadithis maximus to replace Napier grass in some of our production systems. But we also think we have the opportunity to do some crosses. But Megadithis maximus has one major disadvantage, in that only about 2% of the population is actually sexual. So a lot of the materials are apomictic. So what we are trying to do right now in terms of our breeding activity is mutation breeding, because crossing becomes a bit more difficult when you don't have sexual plants. But we are also looking at the 2% that is sexual, to see whether we can do a bit of crossing in that. And as you can see, the number of harvests that we could get is not as many as we get with Napier grass, but it's something that we think would actually start replacing Napier grass in terms of the number of harvests that we can get. We only worked with about 3, we are reporting only about 3 harvests right now, but we have had since then, since when we wrote this paper, another 5 or 6 harvests, and we have started seeing differences in terms of persistence with some of the material. With frequent cutting, some of the cultivars and accessions start dying out, and we will be reporting that data in our future work. We'd like to acknowledge support from GIZ, who have funded this activity, and we are continuing with this work, and hopefully we will be reporting a bit more data in the next sessions. Thank you very much.
Thank you, David. Okay, we have plenty of time for questions. Diana?
Okay. I don't know if you showed first, which is your research station, and how much does it rain there, where these things were evaluated?
Yes, we did. I showed that we did this work in Kakamega. Kakamega is in the western region of the country near Lake Victoria, which receives about 2,000 millimeters of rainfall. But what we have done now is now we have 7 different sites across different agroecologies. That was the preliminary work around Western Kenya, but now we have 7 different sites across the country with the different amounts of rainfall, from very high rainfall areas in Western Kenya to about 500 millimeters of rainfall in Eastern Kenya. And we are now looking at what happens to the different accessions across these different sites. Yeah.
Questions?
Yeah, I was wondering, do you have an active seed industry in Kenya or right now Africa?
Sorry, could I?
A seed industry.
A seed?
Seed industry where producers such as
Yes, we do.
right,
Yes, we do, but a lot of our seed is mainly crop seed, but now we are working with the Kenya Plant Health and Inspectory Services to develop a pasture system where the evaluation and commercialization of some of this seed is starting to come into the system. But just to report, in the 60s, Kenya had one of the best pasture seed systems in East Africa, in Africa actually, but we want to get back into that because right now we are more onto the crop side than on the pasture side. Yes, 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 yes.
Thank you very much for the presentation. I wanted to find out, in terms of adoption, once you have proven that the species can be able to replace anything, what are the chances of farmers adopting this species?
Now, there's quite a huge interest right now of getting pasture into our production systems. And we think it's just the right time to have material ready for farmers because when Napier came and was doing so well, everybody, all the farmers went into Napier. But now Napier with disease issues is not providing any feed solutions for them. So it's just the right time when farmers are looking for alternatives. And right now, bracharia is a big thing in Kenya. Panicums are a big thing in Kenya. So it's just about the right time to get some of these materials to the production systems. And that's why we are working on some of them.
Question?
So we know that East Africa has been plagued by some really severe droughts recently. Can you tell me, has this rainy season that is ending, has it been better in Kenya?
Yes, the current rain season has been much, much better than we have had for the last 6 or 7 years. It's still a very average season, but it has been better than we have had for the last 6 years. So you can see, just like my colleague from Chile showed, we have had almost a continuous drought for the last 6 or 7 years, but this season has been fairly better. And we have seen quite a bit of good performance with some of these materials this year, and hopefully we'll start getting more production. By the way, the genesis of a lot of the materials that we are using from the SEAT gene bank were selected for drought tolerance. So that's why we had interest to bring them into East Africa, because of exactly that situation that we have had. But we have had a better season this year.
Okay, thank you, David.
Thank you.
Okay, we welcome Shelby Grass. She will present the work analysis of a durian-free sorghum hybrid as a forage crop.
Thank you.
Fernando, is there any chance you could use the microphone for your introduction? All that great.
This one.
I don't know what's going on.
This is the.
Yeah, it's just not displaying some of the photos. The other ones seem to be working, so.
Shall I start by this one?
Yeah, it just, there was a photo there, but it's just not showing it. Oh well.
This is the pointer. You go up and back.
Okay.
Do you need the microphone?
No, I'll be able to talk pretty loud, I think. Hi, everyone. Sorry, there was a photo, but apparently it didn't want to load, so. But today I'm going to be talking about analysis of durian-free sorghum as a forage crop. And so most of this work was done for my PhD work. And so to start off with, we're going to just talk about sorghum for a second. Sorghum is a super valuable crop throughout the world. It's really commonly used as a forage crop. It thrives well in semi-arid to arid environments. It's naturally drought tolerant and can perform well in low nitrogen environments. As a forage, it can produce a large amount of biomass, so it can supply a large amount of feed for your animals. And it can have high nutritive value, especially when containing the brown midrobe trait. Although sorghum produces a cyanogenic glucoside called durin, Durin accumulates in high levels in young leaves and stems, and it also can accumulate in different growing conditions, such as frost and extreme drought can cause a durin accumulation. And different cultivars have varying levels of durin accumulation. Why we care about this, though, is durin accumulation in sorghum forages can contribute to hydrogen cyanide poisoning of livestock. So as animals come and chew on the leaf material, it breaks up the cellular separation of durin and durinase, causing the breakdown of durin into hydrogen cyanide. Hydrogen cyanide causes the animal to not be able to utilize oxygen, leading to convulsions, staggering, and death within minutes in severe cases. So generally speaking, farmers manage hydrogen cyanide toxicity issues through different steps. We wanted to take a different look at this. Durin has a pretty simple biosynthetic pathway. It's just 3 enzymes, takes tyrosine to durin, and so we wanted to see if we could create just a durin-free sorghum. And through EMS mutagenesis, we were able to mutagenize the gene for the first enzyme. The CYP79A1 enzyme is inactive, creating a durin-free sorghum. This trait has been incorporated into sorghum lines, and as we're thinking about this, we're thinking this could be a really great benefit for farmers. Because what this looks like is, on the left here, these are plant tissue samples, and on the right, this is a paper treated, it's called a fecal anger test. If the plant tissue is releasing HCN, it turns blue. So our durin-free lines do not release any hydrogen cyanide. So this could be a really great management strategy for hydrogen cyanide release in sorghum cultivars if that is a concern. And so we wanted to look at this in different aspects of forage production. This work was done at Purdue University, and one of the big concerns there is frost. Frost, everybody worries about when we need to take the animals off. We wanted to see how palatable this product is going to be, and then also if animals are even going to perform well on it. And so, well, like I mentioned, frost is a big concern, especially in the upper Midwest. And so as a frost is occurring, farmers need to be conscious about removing their animals. They should remove their animals for up to 1 week after the frost occurs. This is a rolling suggestion. If another frost occurs within 3 days, you have another 7 days to wait until the plants are completely killed off. And so we compared our durin-free hybrid to multiple conventional hybrids at 2 locations, West Lafayette, Indiana, and Colby, Kansas. And what we saw in our conventionals is we saw what we expected with durin accumulation. Day 0 is when the day before the frost occurred. Day 3 is 3 days following and 7 days later. So at our West Lafayette, Indiana location, we saw our conventional hybrids in the darker bars accumulate durin at 3 days, but by 7 days, they'd return to the original levels. This was kind of the perfect frost conditions. We had 1 frost, temperatures regulated again to higher amounts, so that was able to stabilize again. Where at our Colby, Kansas, we had a continual increase in durin accumulation in those hybrids because we had multiple frosts during that time frame, really showing how important that rolling suggestion is, and it's not just a standard 7 days wait. More importantly though, our experimental hybrid is that durin-free hybrid I mentioned, and we saw no effects due to the frost in our durin and it reverting back to durin accumulation or anything. So these should be safe, and so we wanted to take it 1 step further, and we allowed animals to graze going into the fall as the frost is occurring. While we're doing this, we're using that fecal anger test to monitor for HCN release to make sure these animals are safe. And so we had a frost occur on the night of October 26th, and we had conventional hybrids on the side to just monitor HCN in those, but our durin-free, where the animals were, remained HCN-free the entire time. And so this is going to be a really safe option. We did have 1 little blip, but we think that was just a planting error because it was when we were transitioning from our conventional to our durin-free. And so we're like, okay, this is great. All right, animals are safe. They can graze it well, but are they even going to eat it? Does the animals like durin-free sorghum? So, because if the palatability is affected significantly, it could also affect performance. And so we used our durin-free compared to 3 conventional hybrids in a free choice test, where we had our pre-grazing samples to be able to test kind of the yield before And that's in the lighter blue. The darker blue is our post-grazing samples, and this is to show how much the animals were grazing. So to start off with, our Duran-free our yields were consistent with our other 3 hybrids that were there. But if you notice the darker blue bars, they had the most graze. So our post-grazing was significantly less. Our Duran-free was grazed considerably more at 37 and 47% in cycle 2 and 3 in 2019 and 2020. This is 20% more than most of our other hybrids on there. And to show this kind of a little bit more in a quantitative look to see what we were seeing in the field, we also flew a UAV during this time to see this regression using NDVI. And so everything in white, we use the thresholding method, is green plant material. The black is background noise, soil, etc. And so in our red boxes is our Duran-free. This is day 0 before the sheep were allowed, and it's pretty consistent across the field. Within 24 hours, we were seeing the Duran-free being grazed considerably heavier than the others. And by day 2, there's not really any leaf material left. What this looks like in the field is basically just some stalks left in the field, while this is the point where most of the animals started to go for another feed source. And so we're like, okay, great. The animals like it. They're safe. Will they perform on it now? Because at the end of the day, farmers and producers are not going to want a product that their animals are not going to be able to perform well on. And so we compared it to an S&W SP 4105 hybrid, which is the sorghum Sudan grass brown midrib headless variety, compared to our Duran-free hybrid, which is a sorghum Sudan grass brown midrib 6, carrying that Duran-free trait. And so we did a performance with stocker cattle, and based on their weight gains. We allocated 24 cattle to 6 plots, so there were 4 calves in each of the 6 plots, and they were separated based on weight, sex, and sire. And then we had 2 weigh dates. We had a weigh date at the beginning of the experiment, and we had a weigh date at the end. And there was actually 2 at both time frames to get an average. Our field was a 12-acre field, which is about a 5-hectare field. And as you can see, we have our 6 plots that were divided into 4 cells, so we can rotationally graze these to keep them on there for 62 days. Before each of the moves to a new cell, we also took biomass to see how the yields were doing with that regrowth and being able to keep up with the conventional hybrid. What we saw with our biomass is we saw consistent yields compared to our Duran-free, compared to our S&W hybrid that we were using. There was no statistical differences, even though we did see a numerical difference. I want to point out both of these were in the vegetative growth stage at this point, since our other hybrid was a headless and never headed out, while our Duran-free did. So we wanted to just make sure we were comparing oranges to oranges and not apples to oranges at this point. And so we also took some measurements for nutritive quality at this point, and again, trying to keep it more in that vegetative growth stage. And what we saw is our Duran-free hybrid had lower ADF and NDF compared to our SP 4105 hybrid, and it had higher ruminant digestible protein and overall higher total digestible nutrients while it was in that vegetative growth stage. When it started to enter the boot, a lot of our nutritive differences kind of went away, except we still did see a lower NDF at that time. And overall, what we were really looking to see how this was going is our weight gains. So we saw on average a 0.94 kilogram per day weight gain for SP 4105 and 0.92 kilogram per weight day for the Duran-free, which was significantly similar to each other. So we were very happy with seeing that the animals were able to perform well on our Duran-free compared to that conventional hybrid, even though the conventional remained in that vegetative growth stage the entire period. And so overall, we saw that our Duran-free sorghum did not have any Duran accumulation or prussic acid throughout any of these trials. We always monitored for that. Frost did not affect our Duran-free sorghum. It did not cause any Duran accumulation. Our experimental hybrid was favored by sheep during that time. We also did a trial with neurostogenic lines that kind of suggests that the sheep actually can detect the presence and absence of Duran and or prussic acid. We're not sure if it's the hydrogen cyanide causing it or Duran. And then stocker cattle performed equally as well on the Duran-free sorghum as our S&W hybrid we compared it to. And nutritional quality also was similar. So overall, we're able to determine that dhurrin-free sorghum could be a best management strategy for HC and toxicity concerns in forage sorghum. There was a bunch of people that I could thank for this, but I'm gonna just, Jared Kelly, Scott Kronk, Jason Towers, Tynester Tigers is the old lab group I worked with, S&W for supplying hybrid and funding, Ag Alumni Seed, ARGE, and NSF. And are there any questions?
Thank you, Shelby. Any questions?
Do you expect the same performance across but with severe drought?
Yeah, so we did do drought trials as well during the time, and we did not see any dhurrin accumulation at that time either. I actually should say hydrogen cyanide release because we were doing FA testing at that time, but yeah.
In early stages or just late?
We did stay green, so post flowering drought stress period is when we did it at that time. So.
Derek?
Have you seen any difference in seed production potential of either of the dhurrin-free?
So they did some work on that last summer. I don't know for sure what their results were on that. Just from looking at it in breeding plots and things like that, we did not see anything that way, but they did some more work on that this last summer, so hopefully there should be some more information soon.
Alan?
I presume you're accumulating tyrosine. What happens to the tyrosine that isn't being converted to?
That's a great question. So we did some trials with seedlings at one point, and our seedlings were actually bigger in comparison to our non-dhurrin producers were larger than our dhurrin producing. So it could be going into growth. I know there's a similar mutation. It's not the same mutation that's in Australia, and they're showing a little bit of different results. So I think there needs to be some more research done on what's really happening there. They saw nitrate accumulation, but we do not see nitrate accumulation higher in our hybrid either. So I think there needs to be some little bit more biochemistry work to understand what is happening to the tyrosine at that point.
Time for one short question, last one.
Do you think you can expect the same mutation in the Sudanese types?
Yeah, I think it. So this was a sorghum seed grass. So the female, I think, was a Sudan type. So you could be able to make that transition over into Sudans.
Okay, thank you very much. Well, we welcome Wahid Hossein. He will present the work Cytological and Agronomic Evaluation of Interspecific Hybrids between Trifolium repens and Trifolium occidentale. I don't know why does that happen.
Yeah. Okay, and slideshow. Where is this
pointer?
This is the pointer.
Forward, backward.
Okay. And this is the slideshow. Can you help me here? Slideshow. Slide.
Here?
And here.
Great.
And hi everyone, I'm Wajid, and oh, this is wrong presentation. What? I opened my one.
Sorry, may I help you? Which one is?
IGC presentation, this one. Hope this one is the same. Yeah. All right.
Backward.
Backward. Right. I think this is the right one. Yeah. I'm Wajid Hussain, and I'm sharing this presentation with my colleague from PGG Ryerson, John Ford, who is, I think, present here. Yeah. We are having a wide range of interspecific hybridization program at Ag Research, where our target species is white clover, and we have crossed white clover with about 7 wild relatives. And this is one of those series of interspecific hybrids. This work is being funded by PGG Wrightson Seeds. And here we included Trifolium accidentale in crosses with the white clover. It's a stoloniferous diploid perennial, which is one of the progenitor of white clover, and it is indigenous to England, Spain, France, Portugal, and Ireland. The species naturally grows in saline habitat, and that was the reason that we thought that it might be having drought and salt tolerance. And therefore, it can be used as a source of gene tolerance for drought. So our program phases of development, I have just categorized those in 4 categories. Number 1, to create the primary F1 hybrid between white clover and Trifolium accidentale, and then analyze the fertility and the chromosome behavior of the 2 species, and to decide that which progeny we should proceed with. The third phase is the population development, which we think that it's a pre-breeding phase, and that builds plant number and proof of concept experiments. And the fourth one is the full-scale breeding populations based on various hybrid progenies. Interspecific hybridization in Trifolium species is always having a lot of difficulty, because normally the embryo does not develop and endosperm abort, and usually we have to adopt embryo rescue techniques. But here we have devised a method where I was manipulating the pli-D level, and we wanted to create large number of F1 seeds. So I chromosome doubled the diploid accession of Trifolium accidentale, and then used those tetraploid version of Trifolium accidentale with white clover, which resulted in large number of seeds, F1 seed. 2X accidentale is not producing any seeds in Vive with white clover. And you have to adopt to embryo culture, and that provide us very limited number of F1 seeds. So the first achievement of this project was that we have created a protocol where we can create large number of F1 hybrids by manipulating the pli-D level of one of the parent. Here you can see that on the right-hand side top, it's a colchicine treated tetraploid accidentale, and then here these are the F1, and we use the satellited chromosome as marker. So white clover has got 2 satellited chromosome, and when we double accidentally, it has got 4. So the hybrid are having 3 satellited chromosome, and that's the first confirmation of the hybridity of the F1 plants. The second phase, where we were evaluating the hybrids meiotically, is that white clover is, we have reported that it's a allotetraploid species between Trifolium accidentale and Trifolium pallescens, but genetically it behaves as strict diploid species, forming 16 bivalents, and with a very stable NFS disjunction of 16 chromosome. These 2 genomes in white clover are from pallescens, which we indicated with PP, and 2 are from occidentale. And the P and O genomes do not pair in white clover as such. There is a genetic mechanism which prohibits the pairing of the P and O genomes, and P will always pair with P and O will always pair with O. So one of the result of our interspecific hybridization is that when P and O genome come to an interspecific hybrid environment, then we get the opportunity to get pairing between P and O genome, and that we have obtained the data where we have confirmed that not only the interspecific, like modern day occidentally genomes pairs with white clover, but also the P genome, existing P genome in white clover with pairs with occidentally chromosome as well, and that provide us more opportunity for introgression between the homologous chromosome as well as the homologous chromosome. Then population development and introgression. So for this purpose, we carry on the F1. I forgot to mention one point here is that the pollen stainability in these F1 hybrid and later generation reach to parental level, and we are getting quite fertile hybrids in our interspecific hybridization program. Okay, now the population development, we cross F1 with the adopted elite white clover cultivar to create BC1, and then BC1 to elite cultivar to create BC3, and shuffling chromosome from F2 to F3 generation, and that provide us an opportunity to introgress the genome of the two species. And then after introgressing them, then we carry on evaluating them both agronomically and cytologically with some proof of concept trial. So here we initially, when we developed these hybrid progeny, we carried out a small experiment where we wanted to see the drought tolerance of the hybrids in comparison to white clover and occidentale, testing those hybrids at 4 different moisture level. The 36 is the full field capacity, and the 13% is the lowest dry, fully dry condition of the treatment. So here we see that the total shoot dry weight in white clover decline to 16%, while trifolium accidentally dropped to 46%, and the F1 and BC1 decline was 27 and 31% respectively. For the root dry weight, all species root dry weight increased as soil moisture fell from 36 to 21, but as the soil dry, root dry weight of trifolium accidentale and its hybrid increased by 100% and relatively much more than white clover. The BC1 hybrid, which are these one, have greater root growth response than either parental species. We had another small proof of concept trial in a sand pit to evaluate some of our BC1 and BC2 hybrids in comparison to white clover. And here we have collected data on numerous above the ground and below ground characteristics, but just wanted to show that white clover is having this baldness effect. Its taproot dies in the 16th or 15th month of its growth, and then it relies on very shallow nodal rooting system. And if the dry environment comes, then it disappears and the stolon dies. Our hybrids are actually having a strong root system, not only the one which is developed from the nodal one, but also the taproot persisted for much more longer than the usual 16 months period of trifolium repens. The second characteristic of accidentally leaf is that it has got 1 or 2 more extra layer of palisade cells. And we have done a small experiment to see that how much water retention and how quickly the leaf dries. So our F2 and BC1 hybrids showed very slow water loss than their leaves as compared to white clover.
Go ahead.
It's a huge data we collected, which I don't have the time to present now, but this is the strong root system of our hybrids, and that shows that this strong root system provides us more persistence for our hybrids. We run an experiment, a trial, and in the field. I have limited time, but here we have compared those hybrids with elite, highly bred white clover cultivars, and some of the BC2 and BC1 were performing better than the elite white clover cultivar. And this trial persisted for 3 consecutive years in very drought environment, and this is the 3rd-year data. The actual significance is the recovery phase. So some of our BC2 hybrids are showing better recovery than after the drought compared to white clover. So in current phase, we have introgress, repens, and accidentally genomes. A large-scale crossing program is completed. We have created a wide array of hybrid family, selected hybrid family for at various genomic composition of the 2 species, and these material are currently being evaluated in multi-site field trials by PGG Rights and Seeds. So I would say thank you to everyone. I just want to acknowledge Grace. Hopefully she might be here. Yeah, she is here. And our quantitative genetist and plant breeder Zulfi, who helped us in the designing of our experiments, and Prue Taylor, who is our technician, field technician. And thank you very much. I'm here to answer any question.
We have time for one question.
Oh,
What about the seed production of the hybrid?
Yes, the seed production in this particular area of crosses is at parental level. They are very fertile, and we are competitive with white clover. Yes, yes.
More questions?
Thank you very much.
Thank you very much.
Go ahead. So I haven't seen the last speaker, Everline Kirua. Her presentation is not here anyway. More questions? So is Everline Kirua here? Because it's the only presentation that has not been loaded into the computer, so she's not here. So we have plenty of time for questions. So when do you expect that we can have a variety for
We can pay for it, Fernando. I mean, I think the answer to that is that we are in the stages, as I said, of multi-site testing with this material, and it's how big is that benefit, and particularly in the it's in the shoulders of the season, not in the middle of the drought period, but in those shoulders of the season, how much advantage does it provide to producers, whether it's dairy farmers or sheep and beef farmers, and really that's the value proposition that's got to be finalised for this, but there's a lot of really good work.
Okay. Questions? Okay, thank you for attending this volunteer session. So after lunch, we will welcome you at the thematic sessions, and then we will have the IGC business meeting. So enjoy your lunch and see you this afternoon. Thank you.